Well, hey AP, hope you're doing well when you watch this video. This is the last one. I'm not covering a few things at the end. We'll talk about them in class, but here is speciation, a huge concept. So without further ado, glasses on, let's go ahead and crooked glasses. Anyways, let's go ahead and get started. All right, so this is speciation. So what we're going to be doing today is life continues to evolve within a changing environment. Describe the conditions under which new species may arise. So in order for evolution to be true, right, we have to have new species come into play. We have to have new species that only mate with each other and produce viable offspring with each other and diverge so much they stop mating with their original population. As a speciation may occur when two populations become reproductively isolated. The biological species concept provides a commonly used definition for species. It states that species can be defined as a group capable of interbreeding and exchanging genetic information to produce viable, fertile offspring that can also reproduce and make more of themselves. We're going to describe briefly the rate of evolution and speciation under different ecological conditions. Punctuated equilibrium is when evolution occurs rapidly. Gradualism is when it occurs slowly. That's just two vocabulary words you need to know. Divergent evolution occurs when adaptation to new habitats results in phenotypic diversification. Speciation rates can be especially rapid during times of adaptive radiation and new habitats become available. So like kind of filling niches. So new habitats can result in rapid speciation because we have a whole new resources and all of that coming to be. Speciation results in the diversity of life forms, may be sympatric or allopatric, and there's prezygotic and postzygotic mechanisms that can maintain reproductive isolation. Zygote is a fertilized egg cell, so that is important to kind of think of that. So before we begin, some different things that you should or would be helpful to go over is how to distinguish meiosis from mitosis, explain chromosomal basis of inheritance, and describe how mutation, gene flow, genetic drift, and natural selection affect allele frequencies in populations. Okay. So mystery of mysteries. In the Galapagos, Darwin discovered plants and animals found nowhere on Earth. And so how did a flightless bird come to live on the isolated Galapagos Islands? It's pretty interesting to think about. And so the DNA sequence, at the DNA level, similarities indicate that the flightless cormorant, yeah, go ahead and have fun with that, is closely related to flying cormorants. Actually, I don't think it's cormorant. Okay, this suggests that the flightless bird originated from an ancestral cormorant species that flew from the mainland to the Galapagos. I mean, even like crazy things like, giant tortoises, it's just unique to the Galapagos Islands. How in the world did they get there, right? And so speciation is the process by which one species splits into two more species. So this is like a node on a phylogenetic tree. So when one species splits into two, the species that result may share characteristics inherited from their common ancestor. And so this is a difference between microevolution, which is changes in allele frequency, that's the definition of evolution, and macroevolution is above the species level. So for example, the origin of new groups of organisms, like us, like mammals, from a series of speciation events. That's what evolution has to lead to be true. So species is a Latin word meaning kind or appearance. So what do biologists compare when they're comparing that? Morphology, physiology, which is how, so this is how things look, this is how things function, biochemistry, chemical reactions, and DNA sequences, which of course that's the main thing when grouping organisms together. So here's the biological species concept, of course in LO in our learning objectives, and it's to interbreed in nature and produce viable, fertile offspring. They do not breed successfully with other such groups. So gene flow between populations holds the gene flow of species together. So for example, similarity between different species. I mean, look at these. The eastern meadowlark and the western meadowlark, crazy similar. You and I would be like, yeah, that's the same bird. But they are distinct biological species. Their songs and behaviors are different enough to prevent interbreeding. We look pretty different as human beings. We have diversity, okay? But we are the same species, of course, due to our ability to interbreed successfully. And so reproductive isolation is the existence of biological barriers that prevent members of two species from producing biological viable fertile offspring. And so they limit the production of hybrids. And so you have to have a barrier to prevent, to lead to speciation. You can't have gene flow because if you have gene flow will not lead to speciation. So prezygotic barriers block fertilization. So this is before, again, a zygote is a fertilized egg. So before sperm and egg come together. So you can see impeding differences preventing the successful completion of mating, hindering fertilization. All of that happens, okay? So here's some prezygotic barriers. You have habitat isolation, temporal isolation, behavioral isolation. So habitat isolation is they occupy different habitats. So you can see right here, an example of that is two fly species. One lives on apple trees and one lives on, blueberry shrubs. So like that's very different. Temporal isolation, species breed at different times of the day. So in North America, the western spotted skunk and the eastern spotted skunk, they live in the same place. They live in a similar geographic range, but they mate. One mates in late summer and one mates in late winter. Behavioral isolation, courtship rituals, unique. The blue-footed boobies, inhabitants of the Galapagos, they mate only after, I smile because you gotta watch the videos of these guys like showing off their blue feet, man. Look at our blue feet. And so high step, the male high steps, behavior that calls the female's attention to his bright blue feet. Yeah, you gotta watch some videos about that. Here's some also zygotic reproductive barriers. So you can have mechanical isolation, so morphological differences prevent successful mating. So this is the way things look. So snails approach each other head first when they attempt to mate. Once their heads have moved slightly past each other, the snail's genitals emerge, and if the shells spiral in the same direction, mating can occur. Okay, but if they spiral in opposite directions, that will prevent mating. Yeah, and so gametic isolation, so sperm in one species may not be able to fertilize eggs of another species. So for example, this is the truth in corals and sea urchins. Sea urchins release their sperm and eggs into the surrounding water. So how in the world, everybody's around each other, but it's difficult for the gametes of different species, such as the red and purple urchins shown here, to fuse because proteins, so genetics, right? Central dogma, DNA to RNA to protein, on the surfaces of the eggs and sperm bind very poorly to each other, so they can't fuse together. And so that's prezygotic. Postzygotic prevents hybrid zygotes from developing into a viable fertile offspring. So a zygote's made, but the hybrid breaks down. Okay, so reduced hybrid viability, which means the hybrid's chances of survival are low. You can see some salamander subspecies, they may occasionally hybridize, but they don't completely develop, and those that do are frail. This guy is just very, very frail and sick. Reduced hybrid fertility, even if the hybrids are vigorous, which means healthy, they may be sterile. Famous example in biology is the mule. Male donkey and a female horse. Mules are strong and robust, but can't reproduce. Hybrid breakdown. Some first-generation hybrids are actually fertile, but when we travel through time, they start breaking down. So, for example, you can see cultivated rice, different mutant alleles. Hybrids between them are vigorous and fertile. So here's the hybrid. Here's the hybrids. But plants in the next generation carry too many of these recessive alleles and are small and sterile. So there's the middle one. And so there's all, I would know these 2, 4, 6, 8. I would know all those. Those are important ways that speciation, reproductive barriers can develop. Okay. And so limitations, of course, we can't apply this knowledge to fossils. And the biological species kind of emphasizes the absence of gene flow, right? We got to prevent gene flow. But gene flow does sometimes occur between distinct species. For example, grizzly bears and polar bears can mate to produce growler bears. So it's not, that's, kind of scary. So that's not, so it's never a one-size-fits-all. Suppose two bird species, all right, so here's a question for you. Suppose two bird species live in a forest and are not known to interbreed. One species feeds and mates in the treetops and the other on the ground. But in captivity, the birds can interbreed and produce viable, fertile offspring. What type of reproductive barrier most likely keeps these species separate in nature? All right, so a few things. Go ahead and pause and answer it, but then pause. All right, and so hopefully you saw right here viable, fertile offspring. So they can produce a zygote. So the reproductive barrier probably is a prezygotic barrier, and since we live in different places, it's habitat isolation. Okay, so speciation can occur in two ways. Got to know these. Star this. Super important. Star it, highlight it. Let's do some sort of shape here. Nope. Awesome. I'll just draw like a star. It's going to be, this is completely a waste of time. But, yep, horrible star. Okay, so allopatric speciation and sympatric. This is all about same country or different country. So allopatric occurs when populations are geographically isolated. They're not near each other. Sympatric occurs when populations are not geographically isolated. So how does that work? And so, for example, right here, you can see allopatric speciation, a population becomes separate. So here's your original population. Everybody can interbreed. Over time, hundreds of thousands of years, millions, possibly, gene flow stops. Okay? But sympatric occurs while we don't have a barrier, yet we still develop into different species. Okay? Allopatric means other homeland. And so gene flow is interrupted. So, for example, the flightless cormorant, of the Galapagos likely originated from a flying species. And they've done DNA analysis on that. Definition of geographic barrier depends on the ability of a population to disperse. For example, a canyon is a barrier for rodents, but not birds, right? Birds can cross a canyon. Trees can with windblown pollen. And so some examples from science is separate gene pools may diverge through mutation, natural selection, and genetic drift. I would highlight that. Mutation, natural selection. So, for example, mosquito fish in the Bahamas comprise several isolated populations in different ponds. So this is allopatric speciation. Mosquito fish populations in ponds with predatory fish have evolved different body shapes from populations in ponds without predators. So you can see right here, ponds with predatory fish. It's streamlined. Okay. Tail's powerful. Ponds without predatory fishes, mosquitoes have a different body shape that favors long, steady swimming. Okay, different factors, different evolutionary pressures in the ponds. And so that led to speciation. Reproductive isolation may arise as a byproduct of genetic divergence. For example, behavioral isolation involved between mosquito fish from ponds with and without predators. Female mosquito fish preferred to mate with males that had body shapes more similar to their own. So this is genetic divergence. We are becoming more different genetically. Reproductive barriers can also be seen in lab. Be ready for a lot of lab questions that are experimentally isolated and subjected to different environmental conditions. So, for example, isolated lab populations of fruit flies raised on different diets evolved to digest their food source more efficiently and prefer to mate when partners adapt to the same food source. So right here, you have the initial population, and they separated them. Some are raised on starch, and some are raised on maltose. Okay. And then after 40 generations, they ran like mating experiments. And what they saw in the experimental group was, you can see that starch and starch, the females preferred to mate with the males on the starch, that was from the same environment, and maltose also. But in the control group, everybody kind of had an equal chance of mating. And so what you can see here is that although this barrier was not absolute, of course, after 40 generations, reproductive isolation appeared to be increasing. Maybe it may have been caused by courtship behavior that arose as an incidental byproduct of differing selective pressures to different sources of food. Who knows? Maybe you smell different when you eat something different. Who knows, right? But this is leading us to believe, okay, well, that is allopatric speciation. It's been observed in nature. A sister species of snapping shrimp, Alpheus, began to diverge millions of years ago when they became isolated by the Isthmus of Panama. So gene flow occurred, but then all of a sudden it slowly formed, and so you have different sister species here. Very closely related, but not the same. Okay, why? Because this formed. Sympatric speciation takes place in populations that live in the same geographic area. So this one's a little bit harder. It's in the same country. And so it occurs when gene flow is reduced through factors including polyploidy, habitat differentiation, and sexual selection. So first off, polyploidy is the presence of extra, let me just underline a few things, choose a better color than that, let's do purple, extra sets of chromosomes due to accidents during cell division. Okay, so like that's meiosis, right, non-disjunction. Polyploidy speciation occasionally occurs in animals, but it's much more common in plants. Strawberries are, octoploids, right? And so what happens is diploid cell division error, now you have a tetroid cell, meiosis, now you have 2N and 2N, so you have possibly a new species happening, okay? At least 5 new plant species have evolved by polyploidy speciation since 1850. You've seen it. And so 3 diploid species of goat's beard plant, Tragopogon, have interbred to produce 2 new tetraploid species. So you can see right here, these guys are originals, and then hybrid species. 2 new species. You can see right here, doubling the amount of chromosomes, 24 now. Many important crops like oats, cotton, potatoes, tobacco, and wheat are polyploids. The wheat used for bread is an allohexaploid, which means it has 6 copies. Okay, so sympactic speciation can also result from the appearance of new ecological niches. No, we haven't talked about ecology, but a niche is what, and let's actually just let me just type in a box here if I can. Let me just see right here. So a niche is like resources and jobs and fighting for the same thing. So how an organism, like what role it plays, all of that. So like resources and role, and competitive exclusion principle states, competitive exclusion principle states that no 2 species at the same time can go for the same niche. One's going to win and one's going to lose. And but if you have a new niche, you have a whole new job. You have new jobs, you have new resources, new spaces to move into. So for example, North American apple maggot fly can live on native hawthorn trees, as well as more recently introduced apple trees. Flies that use different host species are reproductively isolated. Okay. Sexual selection can drive sympactic speciation. For example, different colors have contributed to speciation in... All right, and so you can see that all right, sorry about that, guys. Had a little problem. But cichlid fish in Lake Victoria. So you can see right here what they did. Researchers placed males and females of these cichlid fish together in 2 aquarium tanks. One with natural light. So what are they selecting? They're changing, and monochromatic orange lamp. Under normal light, the 2 species are noticeably different. You can see normal light, very different. Monochromatic, they look similar, right? And so the researchers then observed the mate choices of the females in each tank. And so what they saw under normal light conditions, females strongly prefer males of their own species color. So 2 different species, right? But under orange light, females of each species responded indiscriminately to males of both species. So equal chances of mating. The resulting hybrids were viable and fertile. So this is a prezygotic barrier, right? The researchers conclude that mate choice by females based on male breeding coloration can act as a reproductive barrier that keeps the gene pools separate. Since the species can still interbreed, prezygotic behavioral barrier is breached in the laboratory. Genetic divergence between the species is likely to be small. Recent common ancestor. And so that's just an example of that. So go ahead and go through these questions as well. We'll talk about these in class. And so many questions remain concerning how long it takes for new species to form. Obviously, we're not there yet. Broad patterns in the fossil record and studies of morphological and molecular data can be used to determine the time it takes for new species to form. There's some terms here like punctuated equilibria, which means the apparent stasis punctuated by sudden change, right? So it's like the Cambrian explosion. Like we have tons of change happening out of nowhere. And so it contrasts with the gradual change. And so all of a sudden you can see, here's gradual change, slowly evolving different. But punctuated models, like new species, boom, like something happened rapidly, and then it was selected for in a certain environment, or they mated and stuff like that, and reproductive isolation occurred. You can see that it's very rapidly. Speciation occurs very rapidly in punctuation. Divergent evolution occurs when adaptation to new habitats results in phenotypic diversification. Okay, so here's like the finch beaks, like a lot of different, the beaks are tools, and so they're used for specific, kind of niches, right? They're going for different food sources in different places. Speciation rates can be especially rapid during times of adaptive radiation as new habitats become available. Adaptive radiation refers to the evolution of new species that allows empty ecological roles or niches to be filled. So maybe like an ancestor finch came a long time ago to the Galapagos, and there was a whole new food source that it didn't eat very well. Well, that would be adaptive radiation if there was a mutation that led to the finches being able to eat that food source. Okay, so I gave you some questions. I'm not going to read these for time. Go through these and answer them. We'll go through them in class, and we'll practice tomorrow. Hey, thank you so very much for taking your time and watching this. I know it was an assignment grade, but hopefully you did watch it. I know some people struggle with watching the videos, but it's helpful. I hope it's helpful. And so we'll talk about it in class. Much love in Christ, guys. See you.